Can you see my screen? Okay, questions about last time or anything else?
On the midterm review guide that we got, there was like later chat modules as well, and like one of them, like 10 or 11, the one I have, it's like said to be expanded on later. Is there a newer version floating around?
Of the guideline for the review?
Yeah.
Okay, that was okay for the midterm. I'm not sure if I've updated it. I will check and send it out again.
All right, cheers. Thank you.
Right. We were talking about the hippocampus and memory, and I backed up a few slides just to make sure that we kind of remember where we were. And this is just to remind you of Hebb's idea of the phase sequence, which is basically the consequence of the postulate that synapses are potentiated if the cell A fires before cell B, then there would be a strengthening asymmetrically in the direction of A to B, and that would lead to cell B starting to fire even before or even without, perhaps, in anticipation of the stimulus that normally would drive cell B. And we have this critical point that is implicit in the Hebb-Mar network, that if the inputs stay on for a prolonged period of time, then the synaptic matrix becomes symmetrical because the output at time t becomes correlated with the input at time t minus 1. And in contrast, if the input is changing quickly, then the output at time t is also associated with t minus 1, but now it's different. And so you get the formation of a sequence in the network, and the synaptic matrix becomes asymmetric. And then I talked about phase precession, and in anticipation, I apologize. I didn't realize that I had put this also in the next lecture on oscillation, so you'll get this twice today, but it won't hurt you. The idea of phase precession is that because the cell itself is firing periodically, but the period of firing is slower than the period of the theta rhythm, the two rhythms are coupled in a way that they come in and out of phase periodically. I'll say more about that in a minute, but you can see the consequences that if the firing of cell 1 is moving backwards in phase compared to cell 2, then overall within the theta cycle, a little snippet of the sequence is being preserved in exact temporal order. This means that the input at the end of the sequence is uncorrelated with the input at the beginning of the sequence, or the beginning of the theta cycle. And that means the sequence is induced to change rapidly in time, even though overall, if you considered the so-called place field, it's changing slowly in time. If the spikes over this period of time were random, then cell 2 would fire before cell 1 just as often as cell 1 fired before cell 2, and we would not get a sequence stored. And this storage of sequences is facilitated by spike timing dependent plasticity, which ensures that the forward direction is strengthened while the backward direction may be even weakened. And the prediction is that if an animal runs repeatedly through a sequence, then cells that normally would encode location B are actually starting to fire when the animal's at location A. And that's, we saw earlier that is in fact observed. So again, this results in a time compressed sequence within every theta cycle. And I pointed out that recently it was discovered that sometimes when the animal is running on a maze and the animal stops, you'll get a reverse replay of the sequence, and we don't fully understand the mechanism of that, but it is thought to be important for assigning credit, as it were, during reinforcement. So when the animal reaches the goal, stops to eat, you get firing backwards in time, and that may play a role in enabling the brain to figure out that when these cells are firing, is predicting that reward is going to happen. So you can think of memory playback as occurring, at least in the hippocampus, in a time compressed form. So if we have this matrix of spikes from different cells over time, and we just compress it, then that sort of gives you the general concept. And I'm going to give you now an illustration of compressed replay that occurs in the prefrontal cortex, which is strongly connected to the hippocampal output. And in this experiment, the animal is running on a platform, having to remember to go to different goals in sequence. And actually, they get to learn this very well. It's surprising how many items in a sequence a rat can remember. And this is sort of the average trajectory of the animal over time. And you'll notice that this behavior becomes very stereotyped. So the animal, runs, gets the food, turns around, runs, gets the food, and he follows the same trajectory over and over again. And so a neuron that perhaps fired here would have a distinct temporal relationship with a different neuron that fired, say, over here, even though there's no coupling between those neurons. This is a correlation that occurs, that is induced by the stereotypic behavior of the animal. Is that clear? So if you have a cell that fires at arm A, I can't remember which is which, at A, and another cell that fires over here, they may not be synaptically coupled in any way, but if the animal runs at the same speed throughout the trajectory, cell 2 is going to fire after cell 1 in a repeated and systematic fashion. And so the time base here is seconds, and you can see, and I'm going to actually, in the next lecture, I haven't explained what a cross correlation is, but I think you get the idea that you see this periodic firing of cell 2 in relation to cell 1. Whenever cell 1 fires a spike, if you look before and after, you'll see that there was a spike in cell 2. And that's, again, simply because the animal is running periodically. Here are the spike rasters from a bunch of trials. And in this experiment, the animals slept before running on this track, on this platform. And there's no relationship between the firing of the pairs of cells. During running on the task, there's this complex relationship which has to do with, where the cells fired and how much they fired. But you see these big slow peaks, plus or minus 4 seconds here. And in sleep afterwards, you see that now there is a correlation between the cells. And if you look closely, you'll see that, for this cell, there's a notch at near 0, just before 0. In this cell, there's a peak just after 0. This one is just before 0, but everything's occurring on a compressed time scale. And the other thing to take note of is that in this compressed time scale, the only part of this slower component that's showing up is the parts that are around the peak. It doesn't, repeat over and over again over long periods of time. And that has to do with the brain dynamics that are occurring during this slow wave sleep, which I'll explain in a minute.
I have a question about the highly stereotyped behavior that you were talking about in the neural
Sure.
activity.
Yeah.
Is this pattern of activity the same as what you would expect to see with like habit formation or like muscle memory, or is it different because it's a memory-based task than it would be for a habit?
Well, I don't know exactly what muscle memory is, but if you do something, the rat is running for food, he's hungry, he's learned exactly where he has to go to get food on each segment of the task. And so it becomes very stereotyped because he's dedicated to running the task. Of course, he may stop from time to time and groom, and that would break up this correlation somewhat. But normally, the task is learned so well. Actually, I should have included a movie of this because it's quite striking, and maybe I'll dig one up and show it next time. But yeah, it's basically a memory task, a spatial memory task. It's not a motor memory because we've done experiments where we've rotated the platform, or actually rotated the task. And so it's not that the animal learns that he always has to turn left here. It requires that he knows where he's going.
Okay, thank you.
Yeah.
Yeah, that's perfect.
Okay, so if you take all of these cross-correlograms and plot and organize them as a function of how far their peak is from the 0 point. So each of these rows is one of these patterns now expressed in colors. During tasks, you can see that it shows this kind of relationship where cells that have a peak far from the origin are at the top, and that's far before the origin, far after the origin. You can see in the sleep beforehand, there's not very much correspondence between the two, but in the sleep afterwards, they're very highly lined up. But notice that the time scale on these two things is compressed by a factor of 8. Okay, so that's an indication that the sequences are actually being played back. Segments of the sequence on the order of maybe half a second or less are being played back on a compressed scale. Another way of looking at the same idea is to take what we call a template, which is to take the average firing of the cells that fired during this particular trajectory or some trajectory and organize them in time so that this is neuron number 1, it fired near the beginning of this trajectory. Neuron 15 fired a bit at the beginning and a bit at the end. So they're sort of lined up. And then we take that template. This is, basically a few time bins, and we scan it over the data for the entire experiment, looking for a match to that pattern. So of course, every time the animal runs through this part of the trajectory, we get a match, right? It's not a perfect match because there's variability. And if we plot sort of the strength of the match as a function of time over the task, we see that during the task, there is a very high degree of matching because the template is extracted from the task behavior and the behavior is stereotypical. But if we look in the sleep before and after, there's not much match. But if we take this template and we squish it by a factor of 7 and do the same thing, now we don't see matches during the task, but we see a lot of matches after the task. And so that's another demonstration with a different mathematical or analytical technique that shows that sequences in the prefrontal cortex are being reactivated. So I wouldn't be telling you all this, and I wouldn't have put so many years of study into this phenomenon if it didn't have something to do with memory. And fortunately for us, it's been shown by a number of people that indeed, if you block the reactivation, which occurs in the hippocampus, which occurs during hippocampal sharp waves, you can do that by doing real-time recording and stimulating the hippocampus to induce inhibition. Every time there's a sharp wave ripple, the cellular activity is transiently suppressed. And if you do that, then you get an impairment of learning a spatial memory task. It's not a total impairment, but it's a substantial one. And of course, this is only done for about an hour or so after the task, so there are probably reactivation events that were never blocked. The other thing that's interesting is that sleep deprivation reduces experience-induced spine formation in the cortex. So after training an animal, in this case, it was a motor task, and imaging the actual dendritic spines along branches of apical dendrites of pyramidal cells, what is seen is that without training, the spines get formed over time, but not nearly as much as when there is training. And similarly, there's no spine elimination. But the point is that if you block slow wave sleep, you have a significant disruption of the spine formation. And we discussed this in a previous lecture, and now I'm a little lost here. Hang on a second. Only to show, and I talked about the fact that the hippocampal output gets projected to various parts in the cortex, strongly in the retrosplenial cortex, but also in the medial prefrontal cortex, which can be imaged with a prism implanted on the other side. These people, Adrian Peyrash and his colleagues, showed that recent memory reactivation in the prefrontal cortex is highly correlated with the emission of sharp wave ripples in the hippocampus, which we showed previously is associated with replay of recent memory. So this shows that there's a coupling of hippocampal replay and cortical replay, which is necessary, of course, for the theory of memory consolidation.
I have a question. How are they measuring memory recollection or reactivation? Yeah.
Well, this is a little bit complicated. I don't want to go too far into it. There are various techniques for doing it. I've shown you a couple of them. I've shown you the explained variance method, which is kind of a sort of overall average. That was the first demonstration. And then I just showed you the two ways of exploring reactivation of sequence memory. And what Peyrosh et al. Did was they used a principal components analysis to look at the reactivation of the principal components of the population spike sequences. But you'd have to go and look at the paper if you're interested in the statistics. It allows for a more precise temporal it allows for a more temporally precise assessment of reactivation.
Okay. Thank you.
Hang on just a moment. I need to put up my other slides here. I can't see where I'm going. All right. So coming back to the study that I showed you where there are place cells in the retrosplenial cortex, in this study, what they did was they used voltage imaging, which I described before, in the cortex. This is the right hemisphere of the mouse cortex, and the retrosplenial cortex is around here. And they had a LFP recording electrode in the hippocampus to record sharp wave ripples. And so what you're going to see in this movie here is the activation of the cortex at the time of the sharp wave ripple, which is at 0 time here. I hope the movie plays. Yeah, there we go. Watch up here. Did you see it? So let me back up here. Okay, now this is averaged over many sharp wave ripples. So in the window around the sharp wave ripple, there is an activity of the cortex. This is the sharp wave ripple LFP averaged here. This is the cortical LFP averaged here. But one thing you'll sort of notice is that the cortex actually starts to be somewhat activated even before the ripple on average. Remember, this is average over many, many trials. So what they did was to take the individual trials, and these are examples of individual trials where this is the sharp wave ripple here, and they plotted the average voltage as a function of time relative to the onset or the peak of the ripple. And what you can see is that there are as many cortical activations that occur before the ripple as occur after the ripple. And this is in in contrast to the previously widely held idea that the ripples emerge spontaneously in CA3 and play out their retrieval of recent information to the cortex, where it triggers activation of the cortical memory. So what this tells us is that on some trials, it's actually activation of the cortex, which is preceding the ripple. And that leads us to the following hypothesis. Some pure speculation. This is research that these people are still working on to prove this, but this is the idea that for a recent memory, you get formation of a few items may trigger the ripple, trigger pattern completion in CA3, which will trigger a ripple. And then that ripple will play back to the cortex and retrieve all the missing items and lead to a big cortical transient. Whereas, according to the memory consolidation hypothesis, in the cortex, you're developing long-range connections that are linking information across the modules. And so you can get spontaneous retrieval widely across the cortex, which then plays to the hippocampus and generates the ripple. And so in this case, the ripple precedes the cortical activation. In this case, the cortical activation would precede the ripple. So that's the working hypothesis for that research program.
I have a question.
Yep.
I just need a quick reminder as to how pattern completion triggers the ripple in the hippocampus.
Well, so let's see if I can find the relevant slide to explain that. Okay, so this is the simple model. Remember that we actually, was it this lecture that I talked about the different kinds of plasticity that have to occur up there? Yes, actually, I should go back a bit here. Way back. Okay, so remember that you have to, in order for this sort of indexing theory to work, you have to have rapid synaptic plasticity in the modules in CA3. Okay, you also have the capability of plasticity in the modules of the cortex. So each cortical module can act as a autoassociative memory. And if you have random activity going on in the cortex and one of these modules goes off, it sends its signal up to the hippocampus, which is a partial recollection of a previous pattern. The hippocampus then completes the pattern, and the output is projected back to the other modules of the cortex. So remember that in an attractor memory, you can get convergence of the network to an attractor state. Even with random noise, it will go to the closest stored attractor. And so if you seed the hippocampus with some information about partial information about a recent event, then the hippocampus will retrieve the index, and that will lead to reactivation of the entire event in the cortex. Thank you. That's a good question. Let's see if
I sort of noticed that the ripple, it's shaped like a Gaussian, right? But it goes up and down and slowly
Well,
gets to its max value. Sort of one.
yeah, I think that's sort of an average behavior.
Mhm.
If you look at individual ripples, they're kind of all over the map. And it has to do with, what are the currents that are generating the LFP that we record as a ripple. Even if you record from two different locations, not too far apart in CA1, the ripple, a single ripple will look different at the two locations because it depends on which cells are firing. It's really just a bunch of cells firing in a burst. So we were here. Escape out of that. Come back to here. So we were talking about interleaved replay in the first part of this module, about the fact that in order to avoid the catastrophic interference problem, you have to have episodes in which of replay in which the old information is being replayed, and mixed in with that should be episodes of the new information, so that the cortex is actually essentially replaying its old memories and mixing them in with the new ones to avoid the interference problem. And actually, that's kind of what this idea is about here, that this would be the replay of an old memory, whereas this is recent replay. And so in general, this is the basic idea of the complementary learning systems theory, but there are two fundamental problems with it. One problem is that the cortex no longer has access to the original data, right? So for machine learning, that's a problem because you've got to buffer up all the original data and play it back, and it means you eventually have an enormous memory buffer. And the other problem is that there's just not enough time to reactivate everything. If you had to refresh all your memories every time you learn something new, you would have a hard time keeping up with the data load. And so computationally, there have been solutions proposed for these problems. The first solution was actually proposed at even about the same time the original theory was proposed by this guy, Anthony Robbins, who was is a computer scientist, and he realized that in fact you don't actually have to have access to the original data. I see there's a typo here, I better fix it. That it's sufficient to put random patterns in if the network will retrieve the closest pattern to the random pattern. Right, so you don't actually have to, particularly if you have attractor-like networks, but even in a deep learning network, they have the capability of retrieving the pattern that's closest to the current input, even if the input is random noise. And so that allows for the network. You can think of the learning as some kind of compact function of a more complex data space, kind of an approximation of the overall, this is a sort of a one-dimensional cartoon of a, almost infinite dimensional data space. But the idea is that the brain is learning a compressed or a compact representation. And if you allow for this pseudo rehearsal by putting in random patterns, that allows the old items to be refreshed and keeps the function from getting completely disrupted. Of course, it has to incorporate the new data points. But the take-home message there is that for real brains, and indeed it's shown for artificial neural networks, that it's unnecessary, well, at least partly unnecessary, to store all of the original data. You can use this phenomenon called pseudo rehearsal, is what Robbins called it, and this is the performance of the network with fully interleaved access to the original data points. This is rehearsal on pseudo patterns, and this is with no rehearsal after putting in new data. So it works. So the other problem is there's not enough time. And that problem was only recently solved. And just to remind you that the problem originated from understanding that if you force the network to learn something new without interleaving, it learns it very quickly, but it disrupts the other stored items. Okay, but it turned out, when looking back at the original simulation, that when we tried to train the network with the penguin, the bird and fish classes were largely disrupted, but the plant class was almost not disrupted at all, which kind of makes sense because the plant class probably involves largely different neurons and different synaptic connections. So it was trying to learn something of the same class without interleaving led to disruption of that class, but not to other classes. And that led to the idea that you might only need to interleave exemplars of the classes that overlap with the new information. That you don't have to interleave classes that are completely uncorrelated with the new information. And so that led to the idea of what we call similarity-weighted interleaved reactivation, where if this is a representation of the hierarchical class structure of the knowledge that's in the system, so each of these things is a different stored item, and we want to add a new item to an already trained network, we would add it and the hippocampus would drive reactivation of that. But the only things we need to reactivate are things that are more or less related by their common hierarchical structure. We don't have to reactivate the things over here that are not related. And so I'm going to skip that slide. That was shown in a simulation that Jay McClellan did in 2020. And it was shown that actually that, at least under a lot of conditions, works quite nicely. And so this is our current working hypothesis about, how this similarity weighted interleaved reactivation, or SWIL, which is British for garbage, might work. So suppose you have in the cortex a bunch of stored memories or stored schemas. Okay, and they each occupy their own attractor space in this high dimensional system. And we put something new in. Well, that something new has some similarity to some of the stored items, but not to others. Now, suppose that we, as a consequence of forming that new memory in the cortex, it left on the cells that were activated what could be called an excitability trace. That those neurons which were activated by the new experience have some kind of persistent change in making them more sensitive or more excitable. Their thresholds are lowered. There are known mechanisms for that that occur during learning where there's a suppression of potassium leak conductances. So when those conductances are suppressed, the cell becomes more excitable because there's less weight on the potassium conductance. And so the idea is that because these yellow things are already attractors, and you put random noise in, there's a greater likelihood that these attractors are going to be activated during spontaneous events because some of their cells are just more excitable. And as a consequence, then you will get spontaneous reactivation of the old stuff, and you'll also get hippocampus-driven reactivation of the new stuff, and then you'll have some kind of competitive learning like BCM or other competitive learning rule going on, which will allow the new memory to carve out some space for itself without catastrophically disrupting the old. Of course, it will change it slightly. So that's the current working hypothesis that is being studied as we speak. So just a few other things about reactivation. You know, I've talked about reactivation and memory, but there's a whole new development of thought about this reactivation phenomenon, which occurs not only when the animal is sleeping, but also in rats when they pause, they stop running, they stop to eat, they just stop running. You get the hippocampus emitting sharp wave ripples. As I showed you before, you can sometimes get backwards ripples. But the point is that there is reactivation going on during a task, and I'm going to show you an illustration of this. Now, what you're going to see are dots on there's a rat running this complex track, and the spikes are color-coded from different cells, and so you can you hear? Okay, wait. Turn on. Share sound. Tell me if you hear it now. Okay. So here's the rat, that burst. You hear the burst? That was a spiking event, complex spike, sharp wave. He's moving around. You hear the blip, blip, blip. Those are spikes. You notice that when he stops, there's spikes from a lot of different cells. Okay, so that's during behavior. And now what we're going to show is the actual reactivated, or the, so now they're going to put the spikes where they happened during running, so that you're going to see the kind of prediction of where the animal is based on the spiking. Do you see that? So during those bursts, the hippocampus is actually firing off the complete sequence almost to the goal arm. And so that should lead you to think that, you know, that maybe what's going on is that the hippocampus is trying out different sequential patterns, or, you know, thinking about what's coming up next. There's all kinds of possibilities there for ongoing decision-making. That the animal stops. If he's in an open field, you might get trajectories to different directions and so forth. And that has been taken up by a number of labs, but there's a very interesting recent paper just came out in Neuron where they did a very interesting experiment. They had one of these sort of mazes where the animal has to find a food source that's always at the same location, but the barriers are made out of like jail bars, so the animal can't go through it, but he can smell and he can see through it. And on different days, they rearrange the jail bars. So the animal has to learn new routes. And then they do the same kind of replay or decoding is the word I'm looking for, of position based on the spikes during the sharp wave ripples. And what they show is that these trajectories that are in the animal's head, he's not actually running, he fires a sharp wave ripple here, and the trajectory goes around the barrier. And if, by and large, when the animal is running this task, so this is the animal's actual performance, and these are various different events of sharp wave ripples that they are following allowed routes to the goal by and large. So it's as though the hippocampus is actually kind of thinking about possible different behavioral routes. And that ties into sort of a whole new cottage industry in human neuropsychology and imaging, which is looking at the question of, you know, how the hippocampus helps us to visualize things and plan actions and so forth. Which we won't be covering in this course, but this is not required reading, but for those of you who are interested in the topic, it's recommended. And that's it for the hippocampus and memory. And now we're going to move on in the time left to talking about oscillations. But before I start, any questions about things up to now? I'll give you a moment to think about it.
So a lot of this for the CLS theory,
Yeah,
like a lot of the computational work with like McClellan requires like a point neuron that's a firing rate model and also backprop. Do you think any of that will matter? Like any of that, like, are those all sufficient, you think? Like,
well, so as I've said before, we don't know what the brain's mechanism is for training multi-layered networks. We know that there's a backprop algorithm that is mathematically sufficient to train the hidden layers. We don't yet know how the brain does it, or even if the brain does it. But there's so many commonalities between, you know, what we see in the, you know, how knowledge is extracted from experience in deep learning systems and in the brain, and how they're kind of hierarchically organized and how the complexity of the features increases as you ascend the hierarchy and so forth, that it's widely believed, I believe, that there is some kind of mechanism for training the hidden layers, and we're waiting for, one of you geniuses to come along in the next generation and figure out what it is. Okay. Other questions? Oscillations. What's an oscillation? Well, our typical interpretation of the term means something that fluctuates in time, right? Fluctuates periodically in time. And oscillations in the brain are everywhere. They come at different frequencies. And so when we think about oscillations in the brain, there are what look like oscillations at very low frequencies, less than 0.05 hertz. There are ultra fast oscillations in the 200 to 600 hertz range. So when we think about an oscillation, we're just going to go back to basics here for a little bit. We can think about a function that varies in time. And if it varies slowly in time, that's a low frequency. If the function only varies slightly, it's a low amplitude oscillation. If there's a wide swings, it's a high amplitude oscillation. So we have frequency and amplitude, but we also have to consider when we have more than one oscillator, what the phase of the oscillation is. So phase. Okay, so two different oscillators can have peaks which line up at different relative positions. Okay, so that's the basic terminology, but oscillations come in many flavors. If you look at for example, a simple square wave, that's a periodic function. Sine wave is a periodic function. You can have a ramp function. Those are all, in a sense, an oscillation in the general sense of the term. You can have signals that vary their frequency over time. So you can have a frequency modulated signal where the period of the function slows down and speeds up. And as you know, that's what an FM radio does. You can have a function that has a fixed frequency, but the amplitude is modulated by another periodic function. Okay. If you look at the human EEG and you filter the data appropriately, which I'll explain in a minute, clinicians typically identify about 6 ranges of observable EEG. That is when it's recorded from the scalp. Scalp EEG doesn't normally contain the very high frequencies because they're filtered out by all that sort of bone and skin. This is just an illustration of multi-channel EEG during a epileptic discharge, where some channels, develop these high amplitude periodic oscillations. Other channels do something different, but more or less at the same frequency, different phases, and so forth. And so that brings us to the fundamentals of how we analyze periodic functions. Actually, how we can analyze any waveform at all. So if we consider a simple sine wave, which has a certain amplitude and a certain frequency. And we can make a plot of the amplitude versus frequency. Usually we call the amplitude, we don't measure in terms of amplitude, but we take the power, which is amplitude squared, for reasons which need not bother us here, but its power is proportional to the amplitude. You can think of, a wave on the ocean with high amplitude has a lot of power in it, but the same wave has not much energy in it. Sometimes this is called the energy spectrum, but in any case, for a pure sine wave, you would draw a single point or a single bar on this graph, right? So it has a certain frequency on the x-axis and a certain amplitude. For a different sine wave with a different frequency and amplitude, it might plot over here. But we can add these two waves together to give this more complicated wave here. And if we broke it down, we could see that this more complex wave is actually composed of two pure sine waves with different frequencies and amplitudes. That analysis was developed by a mathematician whose name was Fourier. And so this is called Fourier analysis. Fourier analysis can be used in the forward direction and the backward direction. So you can take an arbitrarily complex function and break it down into a sum of sine waves of different amplitudes and phases and frequencies. Or you can do the flip side. You can create a complex waveform by adding together sine waves of the appropriate frequency, amplitude, and phase. Okay, so that's forward transform and reverse transform, or Fourier synthesis or decomposition. And this can be applied to any wave shape. For the more complicated the wave, the more terms you likely need. But in fact, you can approximate a square wave, which doesn't look like a sine wave at all, with you can get as close an approximation as you like using more and more components of higher and higher frequency. Okay, so you can see that, adding up these components gives you kind of an approximation of the square wave. If you kept adding in more and more high frequencies, it would get closer and closer to looking like a true square wave. And to get a perfect square wave, you would need an infinite number of components. But, it's a good approximation is usually good enough. And this is widely used in time series analysis. So the first application that we all know about is in an equalizer in an audio system. You can tune out the bass. If you don't want more bass, you filter it out. These days, that's done in the following way. I mean, in the old days, that was done with analog electronic filters. But these days, it's done digitally by doing a Fourier transform on the data, zeroing out the frequencies that you don't want, and then doing the forward transform and recreating the data without those frequencies. And there's a whole, you can do a degree in engineering on this. So here's an illustration of a complex signal recorded from an infant rat and passed through a digital filter where all of the high frequencies are suppressed, but also some of the low frequencies are suppressed. So you set that with the sliders. This is, Beaver's Bandbox or some other, audio analysis app that you can get for free these days. And that's the output. If you zoom in on this, you see that this is the raw waveform, that the filtering leaves only the high frequency components, but it distorts the shape of the wave, right? Because it's taken out all the low frequency components. So that's something you always have to think about when you're looking at brain signals, and you read that this is bandpass filtered between this frequency and that frequency. You say, yeah, and what's that done to the wave shape? If I go back and look at the wave shape, is it distorted in some way? And does that matter for my analysis? There are various ways of representing this. Come back to the Fourier transform here. Okay, so imagine that you had a wave or a signal whose frequency was changing in time, so that at one moment it looks like this, and then the next moment it looks like that, and maybe the next moment it looks like that. So you could think of this as a time series. At time 1 it looks like that, at time 2 it looks like that, and time 3 it looks like that. That will give you what's called a wavelet transform, which is basically a graph of the frequency components of the signal as a function of time, where each slice through the graph is one of those power spectra color-coded, right? So this is the low frequency component. This has sine wave components of 40 Hertz. This one has a lot of sine wave components at this time, between 5 and 10 Hertz. And you can kind of see that in the signal here. So that's called a wavelet transform, and that's also a very useful and powerful technique if you understand what you're doing for analyzing how brain signals fluctuate over time. So each of those is a power spectrum. Here's an illustration of the application of that to two rhythms, which if you stick a microelectrode somewhere in the cortex, you might see that the signal actually over time, has two frequency components. It has a 40 Hertz component and a slower component, but in this particular experiment, they took brain slices and sliced them down the middle. There are also other more sophisticated ways of doing this, showing that the superficial cortex is generating this higher frequency gamma oscillation, whereas the region of cortex below the layer 4, where the thalamic input comes in, is generating this lower frequency. So at the same time, different cortical neurons are oscillating with different frequency components. Here's a spectral analysis of cortical population activity during sleep. And if this is frequency here, this is time in seconds. And these higher frequency, 5 to 20 Hertz are called spindles, and this lower frequency stuff is called delta waves or slow oscillations. So that's what you typically see in the mammalian cortex during slow wave sleep, is combination of delta waves, sometimes occurring with spindles, sometimes occurring without spindles. Sometimes you see spindles without the slow component. There are two kinds of spindles. The normal spindles, which are typically seen in humans and also typically seen during sleep in rodents, but some strains of rat have what are called high voltage spindles, which are very big oscillations. This is the raw signal. So this is the cortical EEG. This is the hippocampal EEG filtered for high frequency. So these are sharp wave ripples here. And there's no connection between the hippocampal activity and the cortical activity. But if you look at the cells in the cortex, they're bursting at the peaks of these oscillations. So if you zoom in on one, you see that during one of these oscillations, a lot of cortical cells are firing, and in between those oscillations, the cortex is in a complete downstate. Moment here, I just need to check where I am. Okay, so now what I want to point out is that not all that glitters is gold. And ask the question, when is a oscillation not an oscillation? And you'll see what I mean in a minute. And the example of that is for these low voltage spindles that we were just talking about. If you look at these spindles, I've mentioned this before, they're preceded by a cortical downstate. And then the cortical goes into an upstate and you get a spindle, and then it'll go into a downstate again. But if you look at the interval distribution between these downstates or between the onset of the upstates, what you see is that there's a lot of short intervals, so one occurs right after the other, and there's a lot of longer intervals. And if you plot the interval distribution on a log linear axis, you get a straight line. And if you think about this, if it was an oscillation, you would expect the intervals to be regular. You would expect to see, power at one frequency. Well, you might say, well, there's a lot of low frequency power here, not much else. But if you look at the actual interval distribution, it's got this log linear characteristic. Now here's a simulation. This is what we call a stochastic process. It's not periodic, it's random numbers. There's the MATLAB code for it. Okay, it's just a bunch of random ones and zeros with a certain probability at every moment in time of it being one. That's called a Poisson process. We talked about that in relation to hippocampal place cells being random. If you then calculate the interspike interval distribution, the intervals between all the spikes, you get a lot of short intervals and not very many long intervals. If you plot this on a semi-log axis, you get a straight line. And that's a characteristic of a random process. So those so-called slow oscillations, well, yeah, there might be some power in the low frequency, but they're not a periodic function. They're generated by a random process. Now, coming back to this graph here where we have the downstates followed by upstates, here's a chat window exercise. What do you think would be the random process that leads to this pattern in the cortex? Everybody think about that for a moment and put your answer in the chat window. It's a great exam question. I'll have to think of that for next year. I gave it away already. what would be, put your hand up when you think you've got an answer. One hand. Was that a hand, Eric? No, that was a down. What do you think would be a random process? Okay, well then, whoever thinks they have an answer, put it in the chat window. Let's see what we got. Okay, spontaneous reactivation of stored and old memory. That's great. That's exactly right. That if you're putting random noise in, sometimes it's going to retrieve a pattern quickly if it's close to the pattern. Sometimes it's going to take a few cycles of the network to, process that. And so the randomness of the upstate distribution is coming from some kind of random process, and likely it's not. That's related to memory retrieval dynamics from random noise. I mean, that's a hypothesis. It's as good as any, better than some. Okay, so that's when an oscillation is not an oscillation, but a random process. Good thing to remember. Now we're going to talk for 5 minutes just about a few other types of ways of looking at data that are useful when looking at neurophysiological brain data. And these are what we call the peri-event time histogram, the autocorrelation, and the cross-correlation. So suppose we're recording from a visual neuron and we flash the appropriate stimulation across the visual field and the cell fires. And we do this over and over again. And so this is the firing before the stimulus. The red line is the stimulus and the cell fired a bunch of times. Okay, this is what we call a spike raster plot. Now, suppose we bin this into little bins and we add up all the spikes from over the trials and plot the sum. That's what it looks like. Okay, that's a peri-event time histogram. It's a very simple idea. Okay, it's just, and if you do the appropriate normalization, it gives you a measure of the probability that given the stimulus, there will be a spike at any given time before or after the stimulus. Okay, that's the basis idea for looking at these two other ideas. So suppose now, instead of using a visual stimulus, we stick a microelectrode in a cell and fire that cell, and we're recording the spiking from a different cell. Or suppose we don't even stimulate the first cell, we just record when it fired, and every time it fires, we look at the number of spikes which occurred before and after the firing. Okay, then we get what's called a cross-correlation function. And here are some illustrations of cross-correlation functions for a pyramidal cell spiking. So every time the pyramidal cell spikes, we look at the number of spikes in this inhibitory interneuron before or after that. Here is a pyramidal cell to a pyramidal cell cross-correlation. Here's an inhibitory cell onto a pyramidal cell. Notice the pyramidal cell stops firing. And this is a reciprocal connection between a pyramidal cell and an inhibitory cell. So it's the cross-correlation function is a means of assessing what we call functional connectivity. Have you all heard that term before? Those of you who know about functional magnetic resonance certainly have heard the term functional connectivity. What is functional connectivity? Well, it's just a correlation. They've given it a fancy name because they wanted to get it published in Science, but it's just a correlation. And that's what we're looking at here. But it turns out that when you're looking at single neurons, people have gone in with microelectrodes and confirmed that when you get very short latency peaks in the cross-corellogram, most of the time there's actually a functional synapse there. And if you look at pyramidal cell to pyramidal cell functional coupling, in fact, see, this is, short latency over the course of, just a few milliseconds. If you look at that, what you see is that the peaks are very rarely very far above the background noise. That's because the pyramidal to pyramidal synapses are relatively weak. So a given spike in one cell doesn't very often or isn't very often correlated or just push the other cell over the edge of threshold so it fires. Whereas if you look at the coupling from a pyramidal cell onto an interneuron, they're usually quite strong. So that's the cross-correlation function widely used. And then if we take one step further and take a single spike train and say, okay, for every spike in that cell, what's the probability that it spiked a certain time before or a certain time afterwards? And to do that, we take the spike trains and just slide them past one another. And we take, the one copy of the train and call that the stimulus, and the other one we call the target, and we get a function which shows the probability of given a cell at time t, what's the probability of a spike at time t plus or minus. Okay, so the autocorrelation function is symmetric. Here is a very bursty neuron. If it fired at time 0, it's unlikely to fire again very quickly, but then it goes up very quickly to high probability. Here's a less bursty neuron. Here is a neuron that is firing very slowly. And so the autocorrelation function is used to characterize different kinds of neurons. They typically have fairly characteristic autocorrelation functions. But notice that all these analyses are based on this simple peri-event time histogram concept. They're the same thing. It's just using different phenomena as the target and the reference. And you can do autocorrelation and cross correlations not only for spikes, but for LFP waveforms. You simply take two waveforms and you say, okay, given the magnitude at time t, what's the probability of or distribution of magnitudes at time t plus or minus delta t? Or to say it differently, you take the whole waveform and so you have two different signals. You just take the correlation coefficient between the two signals, then you slide one signal over one data point and take the correlation again. Then you slide it over another, you take the correlation again, and you plot the correlation as a function of how much you've slid the signals past one another. So that's the cross correlation of a periodic signal. And that's all for today. Questions? Next lecture, we have the pleasure of my friend and colleague, Jeffrey Kritschmar, who's going to give a nice introduction to the computational role of neuromodulation. What does acetylcholine, noradrenaline, dopamine, et cetera, do for cognitive function from a computational perspective. You should find it quite interesting, and we'll see you on Thursday. I'll finish up oscillations in the last lecture, and we can start reviewing. Bye.